target population with limited resources. So this brings in the topic of internal and external validity. So what is internal validity? It is the degree to which a study is free from bias and it correctly estimates what you want to estimate. Now, this depends upon your methods section and it also depends upon your subject matter, because a few examples I have given you, like your decision about what will be the period of exposure or the window of exposure you are interested in. So if you say 10 years period, is it too long, and acute exposures effect of acute effects you are missing on, then to make such decisions, subject matter knowledge is going to be very, very important. So your subject knowledge and your method section both have to be robust if you want to prevent any kind of bias and get valid results. So what is true in your study population is captured correctly means your results are internally valid. And internal validity is a prerequisite for external validity. Now I'll come back here once we understand what external validity is. We all know what generalizability is. That is, whatever you have found in your study, you want to apply it to a target population. Now, there are two ways of understanding generalizability. One is the target population which you for which you considered when you were doing your study, or there can be some other population, like you are looking at India and you say, okay, whatever I find, I want to apply to the population of our country. But can you apply your findings to population of some other country? That is also generalizability, and this can happen for biological phenomenon, for something like a drug, or a physiological phenomenon which can occur in all human beings. So such phenomenon you can easily generalize across countries or across populations. But if it is something related to behaviors or which is very contextual, there you have to be very specific about generalizability, that your findings are generalizable only to a specific target population. So the generalizability is the degree to which results of a study are applying to larger populations, the target population, and only if your study is internally valid, there is any point in considering generalizability. Only then can you think of generalizing it to the other larger target population. So your expert judgment, your domain knowledge and robust methodology are important for your study to be valid. If you look at internal validity, what is more important is your methodological knowledge and your method section. If you look at external validity, your domain knowledge comes into play. Yes, because you should know to whom your results are applicable, to what kinds of populations they can be applicable, and both your domain knowledge and your subject knowledge both together will then integrate both these kinds of validity and will make your results more usable. Are you all understanding these concepts?
Yes, ma'am.
Yes, ma'am.
Yes.
Yes, ma'am.
Because if your study is free from bias, you are going to get a result which is internally valid, which you have captured what is truly there in your sample. And then if your sample is truly representative of your target population, you can confidently say that what is true for your sample is true for your target population. If what you are studying are biological mechanisms, you can generalize your findings to other populations. But if you are looking at something which is very contextual, for example, something which is related to behaviors, then you have to restrict to a closer target population. That is what you need to remember here. And making such decisions, your domain knowledge or subject knowledge comes into play. And these are the references. Many students ask me for articles. So these references I've written. One is which can help you in your theory, where all the different biases are enumerated and described. Another one is also describing the biases in observational studies. Then the second one is again the one from where I picked up Bergsonian bias, but it describes other studies also. And this last one is describing selection bias and information bias, two different kinds of biases with many examples in cancer research, which is also very, very interesting. And then I'm sure you know about the dictionary epidemiology. Does anyone know about the dictionary? So I will share that dictionary by today evening, which will also help you to understand the basic concepts in epidemiology, where all important concepts are defined with interesting examples. So if you have any questions, I'll stop you. There is one slide I want to show before I stop actually, because uh, Joyce, are you able to see the slide
No, ma'am.
now? Now are you
Yes,
able to?
ma'am.
Yeah.
Yes.
So what I want to tell the group is that we have covered the first five topics. We have very briefly looked at mechanisms of identifying research questions and appropriate study designs to answer these research questions. And then we have looked in detail at confounding effect modification and bias. Bias, remember, is something which you should keep reading and exploring in studies. And it's not just your studies. If you try and help out people with their study designs, you will come across different biases which are there and ways of preventing them. And actually, we are ending part one of this EpiBio series. What remains in the basic concepts is measures of frequency association, which we plan to do at our department level and post the video recording. And then what we plan to shift on to is qualitative research in the rest of 2025. So in 2026 we may come up with part 2 where we will focus on study designs, because many of you all have requested especially survival analysis and time to event analysis. So those parts we will try and cover in part 2 of EpiBio series. So after this short you know recording which we'll do for measures of frequency association, we hope to start with the series on qualitative research. That is one thing which I wanted to share. Any other questions or suggestions you have, you can comment. Any questions about bias? Because I have gone very fast. There were a few comments I got in the last class that this requires more time and more examples and we need to go in more depth. I certainly agree, but given the time limit what we have, what I wanted to do is just cover all the kinds of bias. I want you to understand how bias is different from random error. Point number one. Point number two is the two main types of bias: selection bias and information bias, and then this two again subtypes, and then how in each study design these different biases can occur. Try and understand a few examples and then think about what are the ways of preventing the bias. So if you have any questions, you can ask me. Any questions? Joyce, are you seeing this getting recorded? Okay, it's getting recorded here.
Yes, ma'am.
Yeah, I just stopped the recording.